Hi everyone, in this video we're going to talk about negative feedback. The three things we need to know about negative feedback are, first of all, we need to be able to describe what negative feedback means. We need to be able to give examples of negative feedback loops. And most importantly, we need to be able to interpret diagrams that show negative feedback and explain what's happening. Before we do that, let's remind ourselves of the big idea around homeostasis. So the big idea with homeostasis is that the body is constantly trying to keep the internal conditions the same. This allows the cells in our body to function, so to carry out their jobs effectively. Very specifically, the enzymes need to be able to be in conditions that are optimum. If the enzymes are not in optimum conditions, they won't work as effectively, and our cells can't carry out their functions. So homeostasis is a way of keeping the internal conditions the same to allow the cells in our body to function and the enzymes to work in the most effective way. This is a little bit like on a seesaw. When one person pushes their side down, the other person will then also push down to try and rebalance it or counteract it. Now, there's something which links really closely to homeostasis, and this thing is called negative feedback. And negative feedback is a way of our body ensuring that any changes we do make can be reversed and set back to the correct level. This is the process for how a negative feedback loop works. First of all, a change will be detected by a receptor. For example, this might be an increase in a blood glucose concentration after a meal. A coordinator will compare that with the set point being the level that the glucose should be at. And if it needs to, it will tell an effector to respond. In this case, if we're talking about blood glucose, the effector is the pancreas, which is a gland, and it causes the gland to secrete insulin. Insulin, in this case, will cause glucose to be taken in by muscle and liver cells, causing the body conditions to return to normal. Once the body conditions return to normal, we don't want insulin to continue being secreted and having an effect. So the coordinator again compares this new glucose concentration to a set point, and it will tell the effector to stop responding, hence the term negative feedback. Negatively feeding back means that you're telling the effector to stop responding. So in this case, the pancreas would stop secreting insulin, and this loop continues So let's check our understanding. From the three options, I want you to choose the correct definition of homeostasis. Pause the video now and restart when you've chosen your answer. So you should have chosen number 3. Let's have a look at how number 3 is different from 1 or 2. So number 1 talks about external conditions, and that's incorrect. Homeostasis is about the internal conditions in our bodies or cells. Number 2 uses the word fluctuation as opposed to regulation. Fluctuation is something going up and down and changing constantly, but we're talking about regulation, so keeping something at a set level. Next checkpoint. Which of the following is not an example of a homeostatic control? Pause the video now and restart when you've chosen your answer. You should have chosen number 3. Number 3 is not an example of homeostasis or homeostatic control because you are voluntarily choosing and making a conscious decision to pull blankets over yourself. All of the others are involuntary or automatic. You are not consciously trying to shiver or increase your heart rate or secrete insulin or increase the volume of urine. These are things that you are not consciously thinking about, whereas pulling blankets over yourself, you are. Next checkpoint. You need to read each of these really carefully and choose the correct sequence or order in negative feedback. Pause the video now and restart when you've chosen your answer. Okay, you should have chosen number 2. Let's compare number 2 with number 1 and number 3 and see why this one is the correct answer. Well, number 1 is incorrect because we start with the coordinator and then we move on to the receptor. That is incorrect because receptors are always the first point in any of these responses. So 1 we dismiss because it starts with coordinator rather than receptor. Number 3 we dismiss because if you notice carefully at the end, it says at the end of this loop is that the effector increases its response. That would not be a case of negative feedback but positive feedback. Remember, negative feedback is when the effector is stimulated to stop responding or reduce its initial response. If this is unclear, you got that one wrong, go back to the start of the video and listen again to my explanation on what negative feedback is. Let's have a look at an example of this. So what the examiner will often ask you to do is they will ask you to apply your understanding of negative feedback loops to specific examples. And those examples could be things that you've learnt about before, like in this case, controlling blood glucose, or they could be completely new examples. And often what they'll do is they'll give you a diagram to refer to. So in this case, we're asking you to explain how blood glucose is controlled by negative feedback using the information in the diagram. So when we answer a question like this, we always start off by talking about the receptor and what it's detecting. So in this case, a higher or increased blood sugar level is detected by a receptor. The next thing we always talk about is the coordinator and what the coordinator stimulates. In this case, the coordinator is stimulating secretion of insulin. So we've started with the receptor and what it detects. We've then moved on to describe what the coordinator is stimulating. And then we always say the effect of this. So the effect of insulin is to lower blood glucose. Now a clue here, or a hint, is that this part here, the effect of the insulin in this case, is always going to be the opposite of what was initially detected by the receptor. So the receptor initially detected a high blood glucose level, so therefore the effect is going to be to reduce that. So far that should be fairly straightforward and very similar to what we've learnt previously about blood glucose levels. Now we need to make sure that it links carefully to this idea of negative feedback. So what you're going to do next is you're going to say how this decrease in blood sugar then negatively feeds back to the effector. So in this case, the decrease in blood sugar is detected and that itself reduces the rate of insulin secretion. In other words, it causes negative feedback to reduce the insulin production. Let's have a look at another example. Pause the video now and see if you can explain how water and iron levels are controlled by negative feedback using this diagram. If you need to, go back and look at my model answer. Pause the video now and restart when you've written your answer down. Okay, let's check our answer. So, if we look at our answer here, we first of all remember talking about the receptor and what it detects. We always start with the receptor and what it detects. In this case, the receptor is detecting lower water levels. Now you could have conversely talked about higher water levels, and if you have, that's absolutely fine. Lower water levels are detected by the receptor. The next thing we always talk about is the coordinator and what that stimulates. So in this case, the coordinator is stimulating secretion of ADH. If you've talked about higher water levels, then you would talk about a decreased secretion of ADH, and that's absolutely fine. Then we talk about the effect of this, and the effect of ADH is to increase the water levels in the blood. And the easy way to remember this is it will always be the opposite of what's been detected. So if you detect low water levels, you're going to want to increase those. And then this is the hard bit. How does this relate to negative feedback? Well, the increase in water levels is detected, which then reduces the rate of ADH, the thing that's causing the water levels to go up. Hence, this being a negative feedback loop. Let's have a look at another example. This this time you're going to think about the menstrual cycle and the hormones involved in coordinating that, and you're going to look at two possible examples of negative feedback. You can choose to focus either on FSH and estrogen or LH and progesterone. Pause the video now and explain how either of those is an example of negative feedback looking at the diagram. Restart the video when you've written your answer. So let's check our answer. If you focus on FSH, your answer should look something like this. The FSH is produced by the pituitary gland. The FSH causes secretion of estrogen, and the effect of the estrogen is to inhibit FSH. Inhibit means to stop or to slow down. So therefore, the rate of FSH secretion is reduced. Hence this negative feedback loop. If you chose to talk about LH and progesterone, then you want to say that LH is produced, that causes the production of progesterone, and the effect of the progesterone is to then inhibit, which remember means reduce or stop the release of LH, hence why this is a negative feedback loop.